Alright, so we're a group of physics and math majors who are really interested in nonlinear phenomena and kind of how that relates to the body especially. And that's kind of what brought us together for this project. So today we're going to talk about measuring dual heart and brain complexity. And you know, the main takeaway that we want to convey here is how kind of a mathematician or a physicist might approach something physiological like the brain or the heart and kind of analyze that computation.
Before we analyze the complexity of these computations, we need to know exactly what it is that is complex. So, in the brain and the heart, signals are sent, and these are actually what we measure in this experience or in this study. And the way these signals are sent is by an electron moving from one place to another place. This electron motion is dictated by a property called voltage. And voltage can be equated to how gravity causes a ball to fall from somewhere to the earth at a certain rate. Well, voltage causes an electron to move from one place to another place at a certain rate. And in this study, we measure voltage as a function of time to measure the activity of these signals that are in the heart and the brain.
Alright, so here you can see two plots of voltage over time. These are displaying heart rates, which an average human heart rate is about 60 beats per minute, or 60 beats over 60 seconds. You can see in the top one that with low heart rate variability, that maps out to about 1 second per heartbeat, which is that time interval you see between the two peaks. Now this bottom image shows a higher heart rate variability, so on average it may still be about 60 beats per minute, but here it's broken down to 0.98, 1.2, 1.3 seconds, etc. So just a quick poll of the audience before we go into more of this. These are two plots of heart rate in beats per minute over time. If you think the top graph represents a healthier individual, please place a 1 above your chest. And if you think it's the bottom graph, hold up a 2. Alright, so what many of you held up is actually this bottom graph, which is a 2. And while the top and the bottom both have the same mean, similar standard deviation, the top graph shows more complexity, more irregularity, and as people, we think that the body is a physiological system. It moves to homeostasis, greater constancy, stability, regularity, but the top graph is actually the healthier person. So, looking at those two graphs, it's hard to say just from the lines, this one's healthier, this one's not. So as mathematicians, what we do is collect a bunch of data, bin it into categories, and then use those categories to analyze it. So this is an example of how we might do that. You can see with the two blue lines that those are groups of healthy individuals under resting conditions, and they show a more nonlinear or irregular pattern. In opposition to that, you see with the heart failure, it's a more linear line, so lower variability. And this has clinical applications for stuff such as cardiac risk, old age and memory loss, depression, anxiety, and kind of inspired our overall questions, which were, does better memory relate to greater signal complexity in the heart and the brain? And do these also relate to better health in general?
The way we went about answering these questions was through this design. We first had an eligibility survey that determined if the participant met our controls. And then they went through a memory testing system that was designed by Vanessa Zubrin for her honors thesis. And so that memory test was basically 22 pictures and then 1 minute of math followed by a verbal recall. And then after they finished that, and there was 9 rounds of that, after they finished that, there was an ECG placed on their body for 24 hours, in which we monitored, and then they came back 24 hours later and the ECG was removed.
So it's worth going over what an EEG and ECG actually are. So an EEG is an electroencephalogram, and its purpose is to evaluate voltages that Hunter talked about earlier in the brain. And so it does that by electrodes that are connected to the scalp. And you can see on this picture here, there are lots of different electrodes at different locations on the brain. And each of these electrodes will actually send back a signal that we can look at. And it looks something like this. So this is a bunch of signals corresponding to different locations on the brain, and we're actually able to analyze these. So an ECG is an electrocardiogram. It's very similar to the EEG in that it measures voltages, but this is actually the heart instead of the brain. And it does that through 3 electrodes that are connected to your collarbones and your rib. And so with this, we're able to actually analyze voltages across the heart. And if we were to look at those as a function of time, it would look something like this. So this might look similar to what Carolyn showed us earlier. This is actually 2 heartbeats, and you know it's worth mentioning that a heartbeat, or your heart beats like 60 times a minute, like she said, and we take this data over 24 hours. So that's like over 86,000 heartbeats, which is way too much to like go over by hand. So that's where our kind of computation comes in, and so we were able to write some code to actually analyze this data.
Okay, so in understanding the way that we kind of like looked and analyzed the complexity of these different signals, one thing that's important to go over first is it's one of the methods we use, is the idea of fractals, which if you're unfamiliar, fractals are basically self-repeating patterns that persist upon magnification. So if you look, for example, at this generated branching pattern, you would think that when you zoom in on a little piece of it, you would be able to kind of flesh it out and break it down into its constituent elements, and start to see the like simpler parts that make it up. But with things that exhibit fractal behavior, what you find is that as you zoom in, the kind of complex nature and patterns repeat themselves. And you see, even if we zoom in again on this zoomed in portion, the pattern again repeats itself. So there's this kind of this persistent complexity as you look closer and closer. And the interesting thing is that this same pattern is found in a lot of physiological phenomenon, such as the voltage signals from the brain or heart rate over time. So you can look here, for example, this is just heart rate graphed as a function of time. And you can see on the scale of 300 minutes, obviously it looks really complex. But again, you would expect, zooming in just naturally, that you'd begin to see the simpler parts. But we find that as you scale down to 30 minutes, the complexity still remains. And again, if you zoom in at 3 minutes, the complexity remains. So it's kind of interesting that the body shows a lot of these fractal behaviors. So this is one of the questions that we sought out to answer at the beginning, and that is, if we know that there are differing complexities in some of these signals, how do we quantify that and break that down into a number that we can clearly understand? And this long algorithm is called the Iguchi Method. It's one of the methods we used. And what it does is it takes the data that we feed in. We specifically use this for the EEG brain data. And it's able to go through the data and give us a calculated value for the fractal dimension, which essentially is a measure of how fractal the data is. So you can see, again, here's just what some of the EEG data looks like. So we were able to take that numerically and run it through code that was written. This is just a little excerpt of some much longer code that runs the Iguchi method. And through this process, we're able to get a number that says, that tells us exactly how complex the signals are, so that we're able to compare multiple. And one of the ways we did that was by creating color maps. So you can see each square represents a different electrode in different parts of the brain. So we're able to visualize how the fractal nature changes across. And we're able to take multiple color maps and make comparisons. Another method we used is approximate entropy. It's another way of measuring how predictable and how much fluctuation is in a certain time series data. So looking back now, with what we've talked about with complexity and signals in the body, which of these, with a 1, 2, or 3 on your chest, would you say is indicative of the healthiest individual? Okay, it looks like most people got it this time. It is the first one, and we can see visually that it appears to be the most complex. But again, our idea is how do we look at this and assign a number to say exactly how much more complex it is, rather than just making an assumption based on sight. And this method of app-in allows us to do this. So you can see that after running the approximate entropy algorithm, which itself is also very long and computerized for us by code, you can see that the value is higher for more complex signals, which makes more sense because they're less predictable and there's more entropy. And in the less complex signals, it's much lower.
So in conclusion, In order to understand these complex behaviors, what we do is we use these mathematical methods to put a number on these values so we can compare them to something else. And what we hope to do is find a connection between this heart and brain data and healthy individuals. And we really do look forward to analyzing our data in the coming months and figuring out what we can have. And a big thank you to Vanessa Zarubin again for designing the memory, and Dr. Steinmetz for training us on the EEG, and Timmy Phillips and Paige Bolton helped a lot under our testing. And thank you to all the professors who offered extra credit: Dr. Reed, Dr. Pittman, Dr. Lefebvre, Dr. McQuiston, Dr. Bostic, and Dr. Sally.
Any questions for this group?
So in my past research, I've learned that low heart rate variability is healthier because higher heart rate variability is found in people who are have chronic stress, and that leads to heart arrhythmias and sudden death due to cardiac arrest. So where are you getting your information that high heart rate variability is found in healthy people rather than stress unhealthy people?
So there are a couple of references on our last page. We based most of our heart rate research on Ari Goldberger and his studies, but most of the data shows that within a certain range, higher heart rate variability leads to less phase locking, less stalled response. It actually helps you respond quicker to changing stimuli in the environment, and makes you a more healthy individual compared to people who are older and maybe have less response time. And depressed and anxious individuals actually have lower heart rate variability, and that there's supposedly a correlation between those. So hopefully that helps answer.
And do you think that your subject pool would be chronically stressed since you use students, and that their heart rate would be different than the general population?
That'd be something interesting to look into. But we did have certain controls based on our eligibility study, where we studied people with lower stress, without depression, anxiety, and they did fill out two forms: the Becker Depression Index, Becker Anxiety Index, so that we could kind of gauge their stress levels and their general health patterns.
Can you tell us what the if there's a signal, say a flat line or a sine wave, it's totally repeatable, what number does that give you on your complexity scale? And what's the other end? Is there a top end of the complexity scale? I mean, a signal is totally random. What number would it have?
With the approximate entropy, I'd have to look back at the paper to see the exact numbers of it used, but I believe an approximate entropy of 0 would be a completely predictable, like a sine wave, it's interesting. It's not necessarily how it changes, because the sine wave looks more complex than a straight line. But what approximate entropy is looking at is predictability and fluctuation. So even though it's not completely flat like a straight line, it would still have no entropy because it's completely predictable. And it basically looks at the kind of patterns that arise. So a more complex, harder brain signal, you would not be able to predict, or it'd be harder to predict. So it's basically a measure of the patterns. So something like that would come out as 0.